friends, welcome in another video tutorial. So this will going to be the second video of our video lecture bacterial genetics. So this from this video we will going to talk about one kind of horizontal gene transfer named conjugation. So let's discuss. So in the previous video I have already told you that horizontal gene transfer is a transfer of gene between two independent organisms, right? These two organisms do not have any kind of parent progeny relationship. Okay, so in conjugation there is a transfer of plasmid, okay, which is known as aplasmid. Okay, so the aplasmid is transferred from one bacterium to another bacterium with the help of a pilus named conjugation pilus. Okay, so this aplasmid will be transferred from that conjugation pilus to another bacterium that usually lacks aplasmid. So the bacteria which already contains the aplasmid is known as the donor bacterium, and from the donor bacterium the plus is transferred to another bacterium, and this another bacterium is known as recipient bacterium. Clear? So that is what I have written here. Conjugation in bacteria is a process by which plasmid, which is aplasmid, are transferred by themselves alone or along with other DNA elements. We will going to talk about this. We will going to talk about what do you mean by A prime plasmid, what do you mean by HFR strain, all of this, and then you can understand what I actually mean to say by along with other DNA element. Okay, for now just remember that those kind of plasmid can transferred by themselves, okay, either alone or they can also transfer other DNA element, which is most often the part of the donor bacterial chromosome. Okay, that means along with that plasmid some bacterial DNA from the chromosome is also transferred from donor cell to recipient cell. That is the idea. Okay friends, so the cell which transfer plasmid is called donor, and the cell that receive plasmid is called the recipient. So the cell which has received the plasmid from the donor is called transconjugate. That means the recipient cell is also known as transconjugate. Okay, and the phenomenon of the conjugation in bacteria was discovered by Lederberg and Tatum in 1946. So that is the idea friends, that conjugation is a horizontal gene transfer where a plasmid is transferred by themselves alone or with other DNA element from donor bacterium to the recipient bacterium. Okay, and the recipient bacterium usually lacks a plasmid. Clear? So there are two type of plasmid here we will going to talk about. One is self-transmissible plasmid and the mobilizable plasmid. So self-transmissible plasmid are what? Self-transmissible plasmid. So first thing is that the plasmid is nothing but DNA, right? They are extra chromosomal DNA. Okay, so those self-transmissible plasmid have all the genes necessary for their transfer. Okay, not only that, they can also ensure the transfer of other DNA element and mobilizable plasmid. Okay, so the cell which possess self-transmissible plasmid are called donor cell, and other cell which usually do not possess self-transmissible plasmid are called recipient cell. Okay, that means that self-transmissible plasmid from donor cell is transferred to the recipient cell. Okay, and I hope now the concept of self-transmissible plasmid is very clear to you, that self-transmissible plasmid itself can transfer from donor cell to recipient cell. Along with that, it can also ensure transfer of other DNA molecule and sometimes mobilizable plasmid also. Okay, so this type of plasmid contain two type of genes, TRA genes and ODT side. We will going to talk about this. So this type of plasmid are known as aplasmid, F factor or conjugative plasmid. Okay, and now let's talk about what are the mobilizable plasmid. Okay, so mobilizable plasmid are plasmid that contains the gene necessary for their transfer, but they cannot be transferred by themselves. Okay, That means they contain all the genes that ensures their transfer, but they cannot be transferred by themselves. For their transfer, the presence of self-transferable plasmid is necessary. Okay, so mobilizable plasmid is not transferred by itself. I have just told this. Now it requires the help of self-transferable plasmid for its transfer. So that is the today's video. So today we have talked about the basics of conjugation. Okay, it is horizontal gene transfer where a plasmid or self-transferable plasmid is transferred from one bacterium to another bacterium, either alone or along with other DNA elements. Okay, so that's it for today. So I hope this video will be helpful for you. Thank you.